Okay. So the last time we kind of ended on the trip operon and the way that you can negatively control the trip operon. So because tryptophan is so expensive and costly to the cell to produce, if you have high tryptophan present, you want to be able to turn off the production of the tryptophan genes, okay? So the structural genes that synthesize tryptophan. And in this case where you don't have any tryptophan, of course, you don't want that repression to occur. And that is where you have removal or the inability of the APO repressor, which is the repressor, but just in its inactive form, to be able to bind to the operator. And high tryptophan, this APO repressor then forms this dimer and become active and can bind to this operator region and repress transcription. So is there negative control, another way to say negative feedback loop? I don't want to say it's the same way because there could be, depending on the mechanism in molecular biology or gene regulation we're talking about, they can be viewed as differently. So the question is negative control another way to say a negative feedback loop? They're independent depending on the mechanism or the gene regulation pathway that you're looking at. So I don't want to say they're the same. Yeah. So they're a little bit different concepts. So again, so this is usually what's happening. But the problem is that with tryptophan, it's not sufficient to repress transcription of tryptophan by just switching it on and off, right, with these repressor. You need an extra level of control, particularly for tryptophan, because the normal operator binding to the normal repressor binding to the operator and also the termination signal are really kind of leaky. So you can still you still get a little bit of background expression of tryptophan. And again, it really does cause the cell to produce tryptophan. So you don't want to have that background level. So you want to have another way that you can repress transcription of these tryptophan structural genes. And that's where within this region between the promoter and the start of these particular tryptophan genes, you have an additional region, okay? And this region is what helps to promote an added level of repression, okay? So, and this region is usually very similar to the attenuation that you saw previously with termination of transcription in bacteria. And in this particular region, again, between this operator and the start of these genes, you can find a tool kind of a structure. So there's a trip leader region, and then there's the attenuator region. It's all encoded by this particular this particular transcript, right? So you have a start and you have a stop, right? And this is the leader peptide that's formed. And this region is able to really reduce the expression or the transcription of tryptophan. So in the case when you have low tryptophan, the polymerase is able to read through, and we'll discuss how that happens, can overcome this region and read through this region and transcribe these genes. However, when you have high tryptophan, you have a premature termination. So the polymerase falls off before these genes can be transcribed. And that's due to what's happening here in this attenuator region, okay? And the way that this termination works is that, as any termination signal that we discussed previously, you need to have an inverted repeat and a string of T's, right? And that depending on the mechanism, because we talked about row dependent and row independent mechanism. But the idea is that you have an inverted repeat, and the inverted repeat is able to destabilize the transcript with the DNA and release the transcript, okay, and polymerase falls off. So with this attenuation mechanism for tryptophan, it's very similar. It's just, is there a hairpin structure that's going to be able to form? And also, will you have this string of T's adjacent to this hairpin structure? That region within the promoter and the start of the tryptophan genes can form two different types of structure, okay? So you can either have a structure where there's two hairpins by a string of T's, or you can have a structure where there's just one hairpin structure forming. And that's, of course, not immediately preceded by by a string of T's downstream of it. Both of these structures are available within this region that can be formed. What determines which one will form really depends on the state of tryptophan in the cell, okay? So this region, as I mentioned, have an amino acid that you start methionine, you have your stop codon. However, within this region are two tryptophan back to back. This is very unusual, as it should be, because tryptophan is a very rare amino acid, okay? So the chance of you having found a tryptophan the chance of you finding a tryptophan within the cell is like you will find it one every 100 amino acids, right? So it's very rare. It's because it's highly costly, right? So the fact that you have two tryptophan really creates a problem for the cell to have them back to back, right? So we have to make both of these amino acids at the same time. So because of this, in the cell, you have that attenuation region. So if for some reason this sequence or part of the sequence, which encode both of these tryptophan, is available, it will form this hairpin structure. So 1 and 2 will form one hairpin structure, and that the other hairpin structure where there's similarity in the sequence will be formed between 3 and 4, which you don't really necessarily see it here, okay? Between 3 and 4. The same sequence, for whatever reason, as you will discuss, probably will not be formed, right? So instead you have a hairpin structure being formed between the region of 2 and the region of 3, okay? So if this hairpin structure formed, then there's no way that this can really fold back and base pair with this, because these bases are no longer available, right? They're here, okay? So this is usually the mechanism of how attenuation works, right? So it's either of these two conformations. In the case where you have high levels of tryptophan and you want to turn off the production of tryptophan, this is the more desirable structure, right? Because you have your hairpin structure, you have your string of T's, perfect, the perfect terminator, okay? And this is the situation if you have high tryptophan, because you want to stop making it. In the case when you have low tryptophan, this is the structure that's a little bit more desirable, right? Because you want to be able to continue making tryptophan, so you don't want to have this inverted repeat followed by a string of T's being formed. Okay. So then the cell need to find a way, because this is the normal mechanism that will happen within that region. The cell need to be able to find a way to override this attenuation, right? This repression, to override this repression when you need to make tryptophan or not, right? So it really needs to be controlled. So there is the way that you can overcome this attenuation within the cell. So let's say you have tryptophan starvation. You definitely don't want to have this double hairpin structure followed by the string of T's, right? You want to have it in a different structure. And these structures will form because of the nature of the sequence within that attenuation region. So what happens is that when you have low tryptophan, because transcription and translation in bacteria occurs simultaneously, you'll see how it's different when we talk about eukaryotes, right? So as soon as this transcript is made, polymerase comes in and starts to translate immediately, right? So you can imagine that RNA polymerase is somewhere here, right? It's making this transcript and it's there, and then the ribosome is coming in to start translating this transcript into a protein. So because you have low tryptophan, you don't have any tryptophan around, ribosome is going to stall, right? The ribosome stalls because it needs 2 tryptophan and it doesn't have 2 tryptophan, right? So this ribosome stalls on this double pair of amino acid for tryptophan. Because it stalls, it prevents this region of this transcript to form this hairpin structure, okay? So this first hairpin structure is unable to be formed. So what happens, it stalls, the RNA polymerase continues on, right? This hairpin structure is gonna form, but there's no string of T's. So it will continue and there's no terminator, right? There's no inverted repeat and string of T's, polymerase continue, translate, and transcribe the tryptophan genes, okay? And this is how you override that attenuation under situations of low tryptophan. If you have high tryptophan, then you really want to go through and turn off or have an added repression, right? So you really want to turn off the transcription of these tryptophan genes. So again, the transcript is made, the ribosome can come in, right? But the ribosome is not stalled here, right? Because it can, you know, there's lots of tryptophan around, it continue translating this tryptophan. And as it's doing that, it will go. There's a stop codon, right? You'll see when we talk about translation, ribosomes will go, they will fall off when you have a stop codon because this is not an amino acid really, so it just falls off. And because this hairpin structure is formed, then this 3 region will base pair with 4. Okay, and this is where you get your second hairpin structure with your string of T's. You have a terminator, perfect hairpin, string of T's, polymerase stops. You are not able to transcribe the rest of the genes. Okay, and okay, so that's kind of the situation. Yes, Sheila.
So do the hairpin structures form after the ribosome has fallen off?
So as soon as the ribosome goes, for this situation where you have lots of tryptophan, yes. When the ribosome gets to this stop codon, so it will keep going, right? Because it's not stalling on the 2 tryptophans up here, right? Because you have lots of tryptophan around, so it can make it, right? It can translate it. So it will keep going, but then it comes to a natural stop codon, which tells the ribosome that's the end anyway of that protein, that leader peptide that it needs to make. So it falls off naturally here. So because all of this regions is free from ribosomes, it will base pair with region number 2 and form this hairpin structure.
Okay.
Does that make sense? And because these 2 are formed, that leaves these other 2 regions which naturally wouldn't form here, but these 2 regions are now able to form this hairpin structure and this string of T's.
Okay.
So lots of tryptophan, the ribosome can just fly through, translate this leader peptide, falls off naturally as it should at the stop codon. But then this region is now free to form this first hairpin structure because this first hairpin structure is formed. This is the natural one that will form with the second one.
Oh, so it's just basically based off the bonding between 1 and 2, not necessarily 3 and 4, right? Like 3 and 4, the 3 and 4 isn't what causes the termination of the RNA polymerase then. It's just something that happens after as a result of the RNA polymerase falling off.
So 3 and 4 this hairpin structure is what causes the termination, right? But this hairpin structure can only form, right, if 1 and 2 can make the first hairpin structure. And what forces this first hairpin structure is when you have high tryptophan, so that the ribosome can keep going and doesn't stall to prevent this first and second hairpin structure from forming.
Oh, okay, that makes sense.
Right?
Thank you.
It's really this stalling that's really forcing it into this hairpin structure, which doesn't create the perfect terminator, right? Because this doesn't give you the hairpin, the inverted repeat, and the string of T's.
Okay, yeah.
So this is kind of a way to overcome it.
Okay.
Yeah.
Okay, thank you.
Mm-hmm. Okay. So it's actually, it's 3 and 4. These two, having 2 hairpin structure is way more stable than having 1 hairpin structure. Yeah. So this is usually, not, that's why when you have lots of tryptophan and you really want to turn it off, having this hairpin structure is incredibly stable, right? And it really prevents, represses the transcription. Okay. So that's really all I want to talk about attenuation and how you can continue to repress that tryptophan, the set of tryptophan, the tryptophan operon, okay? And the other way that you can regulate control gene expression by using what we call upstream sequences or sequences or region upstream of their transcription start site are also referred to as riboswitches. So this is a phenomenon that was discovered in bacteria, but now we know that you have these similar types of control centers in other organisms as well. And these are really just small molecules that can act directly on the 5' UTRs of mRNAs. The 5' UTRs, if you guys recall, is the 5' region or 5' untranslated region. So it's transcribed, it's just not translated into a protein. It's super important in regulating many aspects of gene expression. And in this case, it regulates the expression of a gene because there are sequences within this 5' UTR region that can control the gene expression, right? And these regions are located or composed in such a way that when there's an interaction between these small molecules and certain sequences within this 5' UTR, it kind of changes the conformation. And by changing the conformation of that region, it really inhibits the polymerase from binding and transcribing that region of that gene. Okay. In bacteria, riboswitches control about 2 to 3% of the genes that are expressed, and a little bit to a lesser extent in other eukaryotes in general. But it's a control center that was discovered a while ago that we're still trying to figure out more about it and what it's doing in higher organisms, such as humans or fungus, and also in plants. And the way that this system works, to have a riboswitch, and this is really looking at the promoter of the gene that's involved in producing riboflavin, which is a vitamin. And in this promoter region, right, so you have your coding region. This is upstream of that coding region. So this is your 5' UTR region. You have these sequences, and it's listed here as an aptamer and an expression platform, but these are really sequences that create these structures, okay? And this is usually referred to as a riboswitch. This particular situation includes the sensing of a molecule. So if you have a molecule, in this case, some kind of a ligand that can recognize this sequence, this ligand will bind to this aptamer. So it will recognize it, it will bind to this aptamer. When it binds to this aptamer, it's able to somehow change the downstream region, this expression platform, into a different type of shape, okay? And because it's able to change this shape, this entire 5' UTR region gets altered. So the polymerase gets, the binding of polymerase to this region gets affected, okay? And in addition, the binding of this ligand to this region, because this region gets changed, you guys remember when we were talking about the repressors binding to the operator, you saw that was a structural change that was happening. So in a way, this changing of this structure within this 5' UTR creates a terminator, yeah? And this creation of this terminator, again, preventing RNA polymerase from being able to continue transcription, is what really attenuates the transcription of these downstream genes, yeah? So 5' UTR region, structurally, this is referred to as a riboswitch, but it's really a series of two regions, aptamer and an expression platform. Binding of a molecule, a small molecule, to this aptamer changes the conformation, attenuates transcription because polymerase is no longer able to bind, okay? And this needs to be regulated in the case where you wanna not produce riboflavin for whatever reason, right? So you wanna control the expression of this particular operon. And this is just not a way that bacteria uses to regulate the transcription. So just to summarize this part before we get into lecture 8, things that you want to think about is we've already discussed this and we had some of the example. How does the LAP operon function, right? Under what conditions it's turned on or off, and how, right? What is the players that's involved in turning them on, right? Like the repressor, the inducer, etc. How is it negatively or positively controlled? The same for the TRP operon, right? What conditions you expect it to be turned on or off and how. We talked about attenuation. How does it work? And how you might override attenuation, right? I mean, because this is really important to be able to do this under conditions where you have low tryptophan, right? You want to turn that repressor off. And very briefly, what are riboswitches, right? I mean, it's not, I'm not going to test you guys on a whole lot of questions about riboswitches. You probably will have one question, right? Like, what is riboswitches? Just because it's not an area that we've discussed too much in detail. Okay, so before I switched to the RNA for transcription in eukaryotes, I'll give a poll and I'll answer any questions about what we just discussed or even the previous lectures for like a minute or two so we can go on. So since this is just in your mind, we just talked about it and you guys are really pumped up and excited about attenuation, you should be able to think about this question. Can you see the poll?
Yeah.
Okay. Give you a few minutes to think about it. Okay. So whilst it's going, or you're almost there at 2 minutes, someone want to explain it or should I explain it? That means I should do it, okay? Yes. Okay, Shayla, go ahead, Shayla.
I chose that the attenuation of the trip operon for the termination of the transcription occurs with higher levels of trip tRNA because it forms the bonds between 1, 2, and 3, 4, causing the early termination.
Yeah, that's correct, right? So exactly right, yeah. And that's usually, you want to think when you have higher levels of trip, right? You really want to be able to turn it off, right? So if you go through all of these steps, I mean, you know, it's kind of the E, right? Because the control region does not lie upstream of the promoter. It lies between the promoter and the start of the genes, right? And termination of translation occurs at lower levels. Actually, it occurs at higher levels, yeah? And then termination of transcription occurs with lower levels, no higher levels, right? Termination of translation occurs with higher levels of the trip tRNA, and it's not translation, right? I mean, the translation actually stalls, right? So you actually don't have higher levels, you have lower levels, right? So in the attenuation, as Shayla mentioned, the termination of transcription occurs when you have high levels, because then that polymerase can fly right through the 2 trips, keep going, 1 and 2 can form, 3 and 4 can form, and then you have your string of T's. And this is really what you want to do, right? You really want to think about, you want to turn off that trip operon. That means you want to have this attenuation functioning when you have high levels of tryptophan.
Professor?
Yes.
Is it important that we know, like I think I asked, is the region 2 and 3 more stable? Because I think I learned in my microbiology class, like that one, because it was more stable, you know, that was like for like anti-attenuation.
So it's, yeah, so it's not necessarily more stable. So it is, as you mentioned, for the anti-attenuation, right? Because that's the desirable situation you want, right? So this is anti-overriding attenuation, right? This is what you want because you have low tryptophan. You don't want to terminate transcription, right? You want to make more of it, right? So this is what will form. It is not more stable because it's just 1 hairpin structure. If you have 2 hairpin structure, that's a more stable structure.
But the more stable structure will terminate transcription.
Yes.
Okay. So the stability of the hairpin doesn't really have to do with like terminating transcription.
No, not really, no.
Okay.
Yeah.
Okay, thank you.
Mm-hmm. So just what force whether you have 1 or 2, and really whether or not this last hairpin structure can form with this string of T's, right? This is really the key part of the terminator that really happens, even with other types of transcription terminators. Yeah. Okay, so transcription in eukaryotes, my favorite topic. So you guys might get way more information than you need. But I'll try to focus on what's on the slides. So the transcription in eukaryotes is way more complicated than prokaryotes for a number of reasons. The main reason is because in bacteria, both transcription and translation happens within the same compartment because there's no nucleus. And also because you can also start translating immediately when you make this transcript, when you transcribe the DNA and you make this transcript, right? So this can occur very quickly. Compared to in eukaryotes, you have compartments, right? You have your nucleus, which is where all the transcription occurs. And this transcript needs to get out into the cytoplasm where the ribosomes are waiting to be able to translate this mRNA. all of these steps that happens is a way that you can regulate gene expression in eukaryotes. Similar to what we see in prokaryotes, most regulation will occur at the transcription initiation stage, right? Whether or not you make the transcript or not. That's like the critical part where you can actually regulate transcription or gene expression. Can you make the transcript? If there's no transcript, then there's nothing else that will happen after that. But in eukaryotes, there's a range of regulation that occurs at the post-transcriptional level, and we'll talk about that in the upcoming lectures. But after you make that first mRNA transcript in the nucleus, there's lots of stuff that happens before that transcript is even exported into the cytoplasm. There's a lot of regulation that's happening at this export, all right, into the cytoplasm. And once it gets into the cytoplasm, there's lots of regulation that happens even before it starts being translated. Okay, so there's lots of regulation that we will talk about. But in general, we'll talk most, for this lecture, we'll just be talking about what's happening at the transcription level. The promoter is way more complex in eukaryotes in terms of how a gene is regulated. So there's many terms that I would like you guys to get familiar with when you think of the gene structure in eukaryotes. So you have this, imagine that this is a gene, that gene X that you need to transcribe. Upstream of that gene, you have regions within the DNA that's important for regulating the expression of that gene. And the key factor, like bacteria, is RNA polymerase. Here is RNA polymerase 2, which we'll talk about. You need additional regulatory elements. So you need general transcription factors. These general transcription factors are important for getting the polymerase where it needs to be to start transcribing that gene. You also have sequence-specific DNA binding factor that binds to very specific DNA sequences also upstream of that gene. They are recognized by transcription factors, which are different from general transcription factors, right? So general transcription factors, as we'll talk about, not all of them necessarily bind to particular DNA sequences, but these sequence-specific DNA transcription factors do. You also have co-activators and co-repressors, right? So proteins that interact with other proteins to help to activate or repress gene expression. Cis elements, we talked about cis elements and trans elements in the last lecture, but here we're talking primarily about cis elements. So these are elements that are present upstream of the gene that needs to be regulated. And in general, the whole promoter region. The promoter is a little bit more complex and a little bit longer in eukaryotes, as we will talk about. All of these proteins, regardless of where they're located upstream or also downstream, as you can see, this is a gene expression and this is downstream, right? This is the end of the gene. But you also have regulation that occurs here to control this gene. All of these regions, right, they're able to regulate a single gene and the transcription of a single gene by interacting with each other somehow, okay? And we'll talk about some of these interactions. Eukaryotes contain at least 3 RNA polymerase. Bacteria only have 1. And each of these polymerases have very different functions. So they transcribe very different sets of genes. So RNA polymerase 1 is a major polymerase that transcribe ribosomal RNAs and different groups of ribosomal RNAs. You also have RNA polymerase 2, which is the one we will be talking about for the rest of this lecture, or in general in this course, right? Because this is the main polymerase that transcribe protein coding genes, so making messenger RNAs. But it can also transcribe other types of non-coding RNAs. Pol3, or RNA polymerase 3, can also transcribe as a class of small ribosomal RNAs. and also tRNAs, which are important for translation. So all the tRNAs that are made are usually transcribed by RNA polymerase III. And then there's also some other types of small nuclear RNAs, which are involved in RNA splicing, as you will see, and also small non-coding RNAs, which are also involved in other types of gene regulation, either at the transcription level or at the translation level. Eukaryotes also have mitochondria, right? And these mitochondria have their own RNA polymerases that help to transcribe the mitochondrial gene. We're not going to talk about those. In plants, in addition to the 3 RNA polymerases that we talked about, there's 2 additional polymerases that were discovered a few years ago, RNA polymerase 4 and RNA polymerase 5. We have not found these in other organisms so far. And they seem to play a role in small RNAs and non-coding RNAs. So as a way to regulate gene expression either at the transcription or translation level, but pretty much just to silence the genes, to turn the genes off, okay? Similar to your bacteria, where you have this single RNA polymerase with different subunits. Do you guys remember the 2 or 3 key subunits of bacteria RNA polymerase? Yeah, exactly right. Sigma, alpha, beta, and omega, right? So there's, these 4 subunits in bacteria. In eukaryotes, RNA polymerase structure is a little bit more complicated. So there are 12 subunits that are present that make up this RNA polymerase, okay? And they have been found in pretty much all organisms that have been studied. They're very similar. They do very similar things in terms of what each of these subunits do. Some of them share homology between the polymerases. So like this subunit 5 is shared between all 3 of the RNA polymerases that's present in eukaryotes besides the other 2 in plants, right? So you can find they're shared. You also have, they have different sizes depending on the subunit. And they're just really named in this case as being, the name is RNA polymerase. It's called B because initially when these subunits were discovered, they were, the nomenclature was in letters. So it was A, B, and C, and so on. But this B is referred to as 2. So it's really RNA polymerase 2, but because the original scientists who discovered it decided to use the letter, so you often see this as RPB. But they're really RNA polymerase 2 subunit 1 or RNA polymerase 2 subunit 2. Okay. So how were they discovered? So yes. So B equals 2 pretty much, yeah. So how were they discovered? Like everything else, there was lots of either molecular biology or biochemistry to identify what these subunits are. And the way that this was done in eukaryotes is that they use this technique called epitope tagging. And the way that this technique works is that you're isolating all of the proteins or complexes that are interacting with each other. So imagine that you're pulling, if you have a magnet and you have a lot of different components attached to that magnet, if you pull that magnet out of a pool of other things, it will bring along whatever is there with whatever is attached to it, right? And that's the whole idea behind this epitope tagging, right? You're isolating a particular protein, and that's what you see here, this blue circle with this little red tag. So this blue circle, which is RNA polymerase subunit 3, have a tag attached to it. So it was engineered this way, okay? So this protein was taken out of the cell, a tag was added to it, and it was put back into the cell, okay? And then you isolate the protein extract and you use an antibody. So epitope, all epitope tags that are used in molecular biology has an antibody or anti-epitope that will recognize it and pull it out, okay? And you add this antibody to connect with this epitope tag and pull it out. It will pull everything that's associated with it, right? All the other subunits that are attached to it. And once that happens, then you can separate them, right? And I remember we talked about protein gels. Usually you can separate this by adding a detergent because the detergent is going to linearize these proteins and also neutralize the charge. So you can run them on the gel and they will migrate on the gel depending on the size of the protein, okay? So that's exactly what they did. And to isolate all of to figure out actually how many subunits are within this RNA polymerase complex. Okay, they knew that they had RNase polymerase 3 because RNAs, they knew that they had the subunit 3 because that's what they used to isolate the rest of the subunits. Okay, and this is really that gel that you're looking at. So this is a protein gel that you're looking at. And on the left, all these labels, right, are the different subunits that you would expect. At the top, you have 4 different lanes. So they labeled these proteins with sulfur, right, S35. You guys remember the Hershey and Chase experiment. And they also used phosphorus 32. The reason that they use phosphorus 32 is because polymerases gets phosphorylated, and when they get the phosphorylation is required for its function. Again, they knew this information, so that's why they added phosphorus 32 to determine which of the subunits are actually phosphorylated. Okay, so 1 and 2, this is the minus means that there's no tag, right? So you're just isolating all of the pool of everything, so everything is there together, so you really can't determine what is there. And you see you have some background bands, right? And this is just background junk, which nobody cares about. In lane 2, you're adding the epitope tag. So you're pulling out everything that's associated with lane 2 with that epitope tag, which is RBP3. And this is where you have all of these different subunits that you can isolate, and you can see the different sizes that's present, okay? In 3 and 4, it's the same type of experiment, except in this case, they're adding the epitope tag. When if it's phosphorylated, you will be able to detect it when you have epitope tag. And you see that actually RBP1, RBP6 are the only 2 subunits within that RNA polymerase 2 complex that gets phosphorylated, right? So those are the ones that are important for phosphorylation, okay? In addition, this is just another gel, which is very similar experiment, because they had 10 subunits, but later on they did a more detailed purification type of experiment to identify the additional components that were there. They had an idea that there were more components, but they couldn't resolve it by doing this experiment. So they did another experiment, very similar, just added another purification step. And you can see that you have the 12 subunits instead of the 10 that you see here. And this is just a ladder. The M is just a ladder, okay? And so we know that RNA polymerase has 12, the 12 subunits. So what does the minus and the plus mean? So the minus and the plus here you have, in this case, right, the protein is labeled with sulfur 35. In this case, the minus here in lane 1, you don't have an epitope tag, right? So imagine you just isolated all the proteins and just run it in a gel, okay? Here, you isolated all the proteins, you add a tag, and that's what that plus means, that this epitope tag is present, okay? Here, in this case, it's plus minus, meaning that you have a epitope tag when it's labeled with phosphorus to see which of these subunits are phosphorylated, okay? So, you know, and that's kind of transcription. That's kind of the subunits, or the 12 subunits of RNA polymerase 2. What we knew also was that if you look at the genes within eukaryotes and how they're oriented in the cell, you can have genes, right, that not necessarily only organized in on the coding, right, to coding strand, right, the 5' to 3' strand. You can also have information on the complementary strand, but they will go in different direction, right? And you can see that these 3 genes cannot be in the same direction because of the way that RNA polymerase transcribe, right? It's just like DNA polymerase can only synthesize 5' to 3'. So the orientation of this gene is different. Okay, so this gene is going in this direction. So pretty much all genes within the genome can be transcribed regardless of what strand of DNA they're present on. Yeah, or what strand the coding information is present on. Because we're talking about RNA polymerase 2, we will talk about the promoters that are recognized by RNA polymerase 2. We're not going to talk about class 1 and class 2 promoters because, as mentioned, class 1 is a lot of the ribosomal subunits or the ribosomal genes, and class 3 is one group of the ribosomal proteins and also tRNAs. So we're not going to really talk about those in this class, but just know that they're different, right? So their elements within the promoters are different. The structures are a little bit different. The location of the elements are also different, okay? So, but it's not something that you need to necessarily remember. So for class 2 promoters, you have 2 parts of that promoter that's present. So you have a core promoter and a proximal promoter. The core promoter is what recognizes all the general transcription factors and RNA polymerase 2. Together, you can have a basal level of transcription, right? Because it's there, so the transcription start site, it directs transcription. So you'll have basal level of transcription occurring at this core promoter. In addition to the core promoter, you have a proximal promoter. This helps to attract the general transcription factor polymerase, but it also have other promoter elements upstream of the transcription start site that also helps to efficiently transcribe these genes, okay? So these are 2 parts, they just have different types of elements that's present, and their location is a little bit different relative to the transcription start site. Very similar to what you saw in bacteria with the -10, -35 box versus up elements or even further distant elements, okay? And within that promoter, so you have elements that are present within those promoter, right? These are cis-acting control elements because they're physically located upstream of this transcription start site within proximity, okay? So you have your core promoter, which is between the transcription start site and this -30 base pair. You need to have this region present for accurate initiation of transcription. You also have the proximal promoter, which is further upstream, could be between -40 to -200. And then you can also have what we refer to as a distal promoter or distal elements or enhancer or silencer regions. These regions can be very, very far away from the gene that it's regulating. So imagine if it's 50,000 base pairs away from this transcription start site, it needs to get here somehow, right? And the way that this happens is that if it's 50,000 base pairs, the proteins that are bound to this region will literally help to bend the DNA or chromosomes and bring it within proximity of this gene that needs to be transcribed. So there's a lot of regulation that happens further away from the promoter of a gene that's also important for regulating that gene. At the core promoter, within that core promoter, there's many different types of elements. These elements are present in some promoters and not in all promoters. Okay, so you have these BRE, TATA, which is very similar to the TATA box we saw in bacteria, initiator elements, and a range of downstream core elements. It's also present. These elements can be found, you can have 1 to 4 depending on the promoter. Okay, so in promoters, for example, if you don't have the TA or the TATA box present, you will need to have this DPE present, and you'll see why that is later on. Okay, so you need to have at least 1 of these promoter elements within a gene to be able to direct transcription. Okay, so you just need to have that within that core promoters. Promoters for highly specialized genes, right? So genes that are important in very specific functions are usually tend to have this TATA box present within the promoter. Unlike bacteria, right, where you had the TATA box being present at a -10, right, location, in eukaryotes, these class 2 promoters, you have a TATA box that's -25 to 30 base pair upstream of the transcription start site. And you have many promoters that are found without this TATA box present, but they have the other element within the core promoter. These type of genes are usually housekeeping genes, right? Proteins that are important for the cell to regulate pretty much all the biochemical pathways. You can think genes like ubiquitin, which is ubiquitous, it's present everywhere, right? That's an important gene. You can also find genes that are involved in different developmental processes also tend to have promoters where there's no TATA box present. So upstream of that core promoter, you have this proximal promoter, right? So proximal just means near, next to something. And within that proximal promoter, you have more than 1 recognition site for transcription factors. So you can have many different sequences within that promoter, okay? Way more than what you will see, for example, in the core promoter, okay? And that's really depending on the transcription factor families that's present within the genome and what the gene is that needs to be regulated. So these regions in the proximal promoter, binding of transcription factors help to increase the frequency of transcription initiation. And they don't necessarily always directly activate or repress, they just kind of increase the frequency or the chance of this this gene to be transcribed. So the way that they can do that is because they can recruit enhancers or silencers. So they themselves might not be able to directly activate them, but they can recruit these enhancers from somewhere else in the genome or silencers from somewhere else in the genome to help with this process, okay? These elements within the promoter, for the proximal promoter, they don't necessarily need to be in a particular orientation. So they can be facing any direction when you read that sequence of that cis element. However, they need to be present at a particular position. Okay, so position is very important for the elements within the proximal promoter. However, enhancer and silencers, which can get recruited there, right, by the transcription factor that binds within that proximal region, these are position and orientation independent, right? Remember I told you that they can be 50,000 base pairs upstream or downstream from the gene, it doesn't really matter. And they can stimulate or repress certain genes or turn them on, turn them on or off, yeah? They're often tissue specific, and they're often tissue specific because they tend to regulate processes that are happening in specific tissues, okay? So you need to really enhance or repress the transcription depending on what tissue type you might be looking at. And depending on the element, they can act as either an enhancer or a silencer, depending on what protein is bound to it, right? If it's an activator protein, then that element can serve as an activator. If it's a repressor that's bound to that element, then it can be served as a silencer. So you can have a single element, but depending on which protein it recruits will determine whether that element, by its association, acts as a activator or a silencer. Okay. So they can act again through proteins bound to them. They can stimulate transcription, these enhancers. And even though they are most times found upstream of the promoter they control, this is not the absolute rule, right? You can have them being in a different orientation, right? So they're being, the sequence is being read away from the promoter. They can be found downstream of the promoter, but they can still act on this promoter. And you can also find them within a gene, right? So they might be found within introns or within the coding sequence of a gene. So it really depends on where they are. And again, they play a role in tissue-specific expression of genes, right? So if you want to see whether a gene is particularly expressed in, particular tissue in the kidney or the liver or something, right? This is where you will find enhancer elements becoming important. Silencers are kind of the same. They silence gene expression. I'm not going to reiterate this anymore. But, unlike bacteria, where the sigma factor binds to that minus 10 to 35 region, and the up element might bind to that minus 40 to minus 60 region, right? The actual polymerase is making contact with the DNA in bacteria. This is not the case with eukaryotic RNA polymerase. They themselves, the polymerase itself does not bind directly to the DNA within the promoter, okay? They rely on transcription factors to show them where they need to bind, okay? And there are two classes of transcription factors we will talk more in detail about, right? Those are the general transcription factors, which is like the most important. And then you also have these gene-specific transcription factors, which functions as activators, okay? And they really help to recruit the polymerase to the promoter of that gene to be transcribed. These general transcription factors combined with RNA polymerase will form this pre-initiation complex. And this pre-initiation complex, once it's formed, there's additional modifications to the RNA polymerase that needs to happen in order for transcription to actually begin, okay, for elongation to happen. And we'll talk about those steps in a minute. So this kind of a blob that you're seeing here include some of the proteins that are present at the promoter in order for transcription to occur within eukaryotes, right? So you have these general transcription factors and additional, so there's 6, right? General transcription factors, and you have your RNA polymerase 2, all descending on this promoter to initiate transcription. These 6 general transcription factors are key in regulating transcription and recruiting the RNA polymerase. They're important in terms of how they bind and when they bind, okay? So they bind in a specific order to the DNA to be able to recruit the RNA polymerase, right? So it's not like they all just come together. That was the initial thought actually when they were discovered, was that everything just randomly forms this complex and then everything comes and sits on the DNA. But now we know that individual general transcription factors will bind in a particular order, okay? And do you guys know how they figured this out? Well, they had to do some kind of a protein DNA binding assay. And what are the 2 protein binding to DNA assays that we've talked about so far? Yeah, the filter binding assay. And what was the other one that was in the exam with the RNA polymerase in the different regions that was protected? Footprint, right? So DNA footprinting assay, yeah. So that's exactly how they figured out what is happening at this promoter. And so we had the two assays to determine the protein binding assay, which we talked about. So they used a technique called EMSA or a gel shift assay. So either EMSA or gel shift assay is what is commonly used, the terminology that's commonly used in the lab these days. And EMSA just stands for an electrophoretic mobility shift assay. Okay, so you're using a gel to run the products on a gel so you can actually see it. And it shifts if there's protein binding to DNA, you get a shift, okay? So the way this technique works is that you have your DNA that's radioactively labeled, right, as always, and then you have a protein, right? So the red is the protein and your DNA is labeled. You usually run just your DNA that's labeled so you can see that you have your labeled DNA. When you add a protein, the protein binds to this DNA. It's a larger molecule. You get a shift in that band, right? Not everything binds, so you always have some remaining, right? So, but you have a shift, and that's what you're looking for if the protein is binding to DNA. If you add another protein, in this case yellow, again, the whole complex gets larger. You have another shift, right? And this is why this is referred to as a mobility shift assay, because the more proteins you add, the shift, the higher and higher band, larger and larger band you will get, okay? And this was the experiment that was done to show that there is an order of the general transcription factor that binds to the DNA, and also which of these general transcription factor actually bind first to the promoter, okay? So this is exactly a gel you're looking at, right? Labeled DNA that you have, and all of these bands to the bottom are just things that are excess DNA that's labeled, okay, that doesn't bound. What you have, these nomenclature telling you which of the complexes these bands refer to, and on the top of the gel, it shows you what is being added, okay? So all of these are being added, and what's present in that lane, that particular lane, okay? So when we have 6 general transcription factors and the polymerase, okay? So in the first lane, just for reference, right, you have the general transcription factor D and A, right? You can see this complex here, okay? The complex is formed, it binds to DNA, so you get a shift, okay? And in that case, if you were to add B, you see it goes up, right? You're adding another protein, so the shift is getting higher, okay? In this lane, you add another one. So in this case, you're adding D, A, B, and then you're adding F. You don't get a shift. So this suggests that F is really not doing anything, right? It's binding, but it's not really contributing to the binding to the DNA, okay? So it's not really there. However, so when you add this F, you don't get this shift, right? So that means that this F is not somehow making contact with this complex, right? So you still, it's really just the D, A, and B and the DNA, so the band is the same. In this case, when you add RNA polymerase now instead, right? So you have an increasing amount of RNA polymerase 2, the same 4 subunits, you see this band getting brighter and brighter and brighter, right? So the shift is getting brighter and brighter as you're increasing the polymerase. So the more polymerase you have, the more this complex is really binding to that DNA, okay? Now, so this is the 4. So what this tells you is that RNA polymerase is causing this shift, and this actual general transcription factor F only binds and shifts this complex when RNA polymerase II is present. And that is what's emphasized here, right? Because again, the same complex you have, but you're decreasing the amount of general transcription factor F. And you can see when you start decreasing F, you have a decrease in that complex being formed. So when you only have DAB and POL2, you don't get this shift, right? So even though it wasn't important here, it is important. It's just that it cannot bind without RNA polymerase II, okay? And the same RNA polymerase II cannot bind without it, okay? So these two work together. What they also notice in addition on this gel is that when there's no D, nothing binds, right? So there's no shift, you just have the DNA. In the absence of B, you still get some binding. In the absence of A, you still get some binding, okay? So what this told them is that TF factor D, TF2D, is the first factor to bind, because without it, none of that complex can form, okay? And in addition, sorry, yes, Sheila?
Can you go back to the previous slide? So when they remove the D, nothing can form, but then the next one that's minus B, that one still has the D though, right? Or is it continuously taking it all off?
It still have the D. So the only thing that's absent here is B, and the same here. The only thing that's absent here is A.
Oh, okay. Thank you.
Good, thanks, that's a good question, yeah. So based on these type of experiments, they were able to conclude that the transcription factors bind to the promoter in a following order. So TF2D, with the help of A, binds to that Tata box, right? And they form that DA complex as the first complex you saw. TF2B then binds, right? Generating this DAB complex. TF2F helps RNA polymerase bind, right? And then you form this other complex. And then last, you have E and H, which bind to form a complete pre-initiation complex. And we'll talk why H is actually, E and H is important a little bit later. They then showed by DNA's footprinting assay exactly where those complexes was binding within the promoter, right? So TF2D, A, and B binds to this Tata box within the promoter. So again, footprinting assay, what is protected means that you're not cleaved. That means the protein is bound to that complex, right? And if you were to say you have D alone, there's a little bit of binding, right, at that Tata box, right, which is the first one. And you're adding it, the complex is getting larger, so you have more region that's protected. And when you look on the gel on the right, again, no protein, right? Everything gets cleaved, right? Similar to this one. DAB, you have your protection that's happening here, right? When you add polymerase NF to this complex, you get a much larger region being protected, right? It's not just upstream of the transcription start site, but also it covers some parts of that transcription, downstream of that transcription start site, right? There's a huge complex that's sitting on this promoter once you add polymerase and the transcription factor F. Okay? So now they know exactly the location and the order that it's happening. Do you need to know the order? Yes, you do need to know the order of this formation of the complex, right? And which of the transcription factors are the one that actually binds there first and what gets recruited. So this is usually a good way to kind of think about it. This is also in the textbook, right? So you have your DAB complex being formed. D is connecting to that Tata box in the DNA. And then these two gets recruited. Polymerase with the help of TF2F, then gets added to this complex to form this pre-initiation complex. How is TF2D actually binding to this DNA, right? Remember, it's not the polymerase that's actually binding to the DNA, it's this TF2D that's actually binding to the DNA. Within that TF2D, like everything in eukaryotes, it's a number of other subunits, right? So TF2D is not a single, just a single monomer. There's many, many different proteins that's in part of that complex, okay? So it contains several subunits. Some of those subunits is the Tata box binding protein, TBP, highly evolutionary conserved, meaning that it's found in every organism, eukaryotic organism that we've looked at so far. In addition, there's 13 TAFs, which are TBP-associated factors, okay? So this is this complex that's descended down on this Tata box in the DNA. So all of this is TF2D, and these are the components of TF2D, okay? The way that TF2B binds to the Tata box binding protein of that TF2D, right, at the Tata box is via interaction, right? So the C-terminal domain and polymerase, it binds to the C-terminal domain of the TBP and polymerase via the N-terminal domain, okay? And that's how it helps to anchor its position at that promoter. So it kind of provides a bridge in action that's positioned between the 25 to 35 base pair downstream of the Tata box, okay? And that's why you saw that huge region that was protected in that gel, okay? And TF2B really helps to bridge that interaction there. And it plays a role in establishing that transcription start site, right? Because it helps, again, it's there. It bridges the polymerase with the TF2D complex. So it kind of really sets where the transcription starts over the transcription start site where it is. The other general transcription factor that's super important is TF2H, which is the one that gets added last, but it also contains multiple subunits, right? It contains nine subunits that's separated into two complexes. So one of the complex is a protein kinase complex of 4 subunits. And you guys remember kinases are involved in phosphorylation, right? So it has the ability to phosphorylate the DNA. And the other complex is a complex containing 5 subunits. 2 of those are DNA helicases and ATPase activity. So helicases help to unwind the DNA. So you can see, you can imagine that when everything gets added and this TF2H gets present at that promoter, at the transcription start site, you need to start unwinding some of the DNA, right? You have that open complex that needs to be formed, and then you also need to continue to unwind the DNA. And that's where some of these subunits within this TF2H becomes important. TF2H plays 2 major roles, right? Besides what I just told you with unwinding the DNA. But the other function, which is absolutely important, is this phosphorylation, right? And that's where this protein kinase complex comes in. You need, RNA polymerase is usually in an unphosphorylated state. It needs to be phosphorylated in order for it to become functional, okay? And that's where TF2H becomes important because it helps to phosphorylate the C-terminal domain of RNA polymerase 2 to really initiate transcription and elongation, okay? Okay, let me just finish these 2 slides and then we probably have to stop and go because the other next set of slides are going to be lots of information too. So the phosphorylation of the C-terminal domain is important, as I mentioned, for initiation and elongation. So the form that is present when it's unphosphorylated is RNA polymerase 2A. And when it's phosphorylated, it's considered RNA polymerase 2O, okay? And in this C-terminal, what gets phosphorylated is this C-terminal domain of RNA polymerase. And the regions or the amino acid that gets phosphorylated are serines, okay? So in this C-terminal domain of RNA polymerase, so this is the RNA polymerase, usually proteins have directionality, as you guys recall, right? So you have N-terminus And then this is the C terminus, and that's why it's called the C terminal domain, right? It's this tail that's sticking out kind of from the RNA polymerase. Within this C terminal domain, you have the range of different amino acids. This region is formed as a tetrad, right? And this tetrad or heptapeptide has multiple repeated amino acid. So these serines at the 2, 5, 7 position are really important in initiating transcription, elongation, and termination, right? Because they get phosphorylated along the way, and the phosphorylation status will change along the way as RNA polymerase is transcribing the RNA, okay? So these are all, again, different stages depending on which one gets phosphorylated. So I'll pause here so we can go over some poll questions to see where we're at, but we'll talk more about how it gets phosphorylated, the precise mechanism in the next class. Yeah, okay. You guys have any questions? It's super exciting stuff, right? Okay. Okay, let me see. Let's try another. I don't know how far we've gotten. Okay. So we talked about the major difference between bacteria and eukaryotic RNA polymerase. What is that difference? What is that major difference? There's many differences. What is the major difference that we talked about? Okay, we have many different options here, I see. Okay, maybe we can okay, I'll go through these as you guys are answering, right? So one major difference between bacterial and eukaryotic RNA polymerase is that, would you say prokaryotes have, does prokaryotes have several RNA polymerases?
No.
No, right? They only have one, right? So that's not, that's definitely not the case, right? Eukaryotic RNA polymerase can polymerize RNA in the 3' to 5' direction. No, right? That's correct, right? Everything is 5' to 3'. That's for both organisms. Yeah. Prokaryotic RNA polymerase recognizes several different promoters. No, right? This is only because one polymerase compared to the polymerases in eukaryotes, where some are recognized for RNA polymerase 1 or RNA polymerase 3. So eukaryotic RNA polymerase has only one polypeptide. You know that that's not true, right? Because you just saw the multiple subunits for RNA polymerase 2. We have 12 of those, right? And so then the only other option that you have right now is that eukaryotic RNA polymerase cannot read the DNA to find the promoter sequence. And that's exactly what we just talked about the whole way, right? We talked about the fact that you need the general transcription factors to read that DNA sequence and bind to that TAA box and recruit the RNA polymerase, right? That's really important to remember. Okay. So that's with that one. Someone asked if these polls are being graded. I mean, all of these information I'm saving, and it's all going to be counted as extra credit work, right, for you guys. So, and just for the attempt, even if you, doesn't matter, because I'm not going to look to see whether you have it right or wrong, just that you participated. This is really for you. And some of these questions you will see again in the multiple choice question in the exam. I mean, I don't know which ones, but there are going, it's quite likely you will see them again. So that was that one. I really have to go. I have a meeting at 2, so I want to give you guys some time to ask questions, but I have other poll questions that we can go over in the next lecture as well. Any questions?
Thank you.
Thank you. Okay, all righty. Well, have a good day, and I'll see you guys on Thursday. I'll try to get everything sorted out for the exam by Thursday, so we will talk then. Okay? Thank you.
Thank you. See you Thursday.
Bye. See you, Sheila.
Oh, hello, Professor.
Yeah, just 1 second. Let me just 1 second.